In this part, I'll talk about a few important segmentation algorithms and the general principles behind them. So let's start with EM segmentation. So here we're going to assume the following statistical model. Every voxel is going to have a label, which we'll call L, and an intensity, which we'll call I. I'll use Li to represent the label at voxel I, and L underlined to represent the collection of all of those, that is, the labels for the whole image. And same goes for the Ys. Now, these two are going to be called the intensity model, which tells us how to get the intensities from the labels, and the spatial model, which tells us how to get the labels in the first place. That is, what labels go where. So remember that our goal here, we have the intensities Y, and we want to segment the image. That is, we want to find the labels L at every voxel and use those to figure out what class each one belongs to. So for this example, I'm going to use the white matter, gray matter, CSF example we talked about before, but this method can be applied to a wide range of image segmentation problems. All right, so our spatial model, to start out with, is going to be very simple. We're going to say that in order to get some configuration of labels L, every voxel's label Li, independent of every other one, is going to be gray matter with probability, some probability pi gm, white matter with some probability pi wm, and similarly for CSF, probability pi CSF. And these guys are going to add up to 1. And this tells us how to get the labels at every voxel. So notice that this is overly simplistic. We know that, for example, voxels near the center of the brain are more likely to be CSF because they'll fall into the ventricles. But this doesn't capture that at all. And actually, in just a few minutes, we'll talk about a more sophisticated spatial model that actually does. But for now, we'll stick to the simple one. Now, what about the intensity model? Well, the intensity model will assume each voxel's intensity. So the intensity model tells us how to get Y from L. And just as above, we're going to assume that they're all conditionally independent. And each one of these is basically going to be a mixture model. Meaning that if voxel I has label gray matter, then it's going to be random with mean then it's going to have this mean and be randomly distributed near there. If it's white matter, it's going to have mean mu wm, and if it's CSF, it's going to have mean mu CSF. So this is our very simple intensity model and our simple spatial model. So notice that the parameters here are these mu's and these pi's, which we don't necessarily know a priori. Also, we don't know the labels, since that's what we're trying to find. So the EM algorithm, and if you're familiar with the EM algorithm in general, this should look very familiar. So it has 2 steps. In the E step, we're going to fix the parameters and estimate the labels. And intuitively, this makes a lot of sense, because if we knew all the parameters, then for every voxel we can say, well, at this voxel, the intensity is closest to one of these means, and we can weight that against what the probability of it being each one is. So we can easily find out what label goes to each voxel. Similarly, the M step is going to flip it. It's going to fix the labels and estimate the parameters. And these are also pretty intuitive. So for example, the mean of gray matter, remember we want to estimate the parameters, which are these boxed quantities. The mean of gray matter, for example, will be roughly the average of all voxels that are labeled gray matter from the E-step. And we can repeat this until convergence. The EM algorithm has a lot of nice statistical properties. I won't go into detail on that right now. But this is really the big idea behind the EM segmenter. There are a lot of extensions, more sophisticated intensity models, more sophisticated spatial models. And in fact, let's talk about one of those spatial models right now. We're going to use an atlas. An atlas is basically a template that tells us at each location what the probabilities of each label are. That is, it's a smarter way to model the label distribution. It captures the intuition I mentioned earlier about, for example, we know near the center of the image, it's more likely to be CSF. And the atlas will tell us exactly that. Notice that it requires being able to draw correspondences. Otherwise, we can't say that, oh, this spot in the atlas corresponds to this spot in the image we're segmenting. So in order to use an atlas, we're going to have to do registration. That is, we're going to have to draw correspondences, or have to align the images in order to use it. So atlases are useful for giving us spatial information. They help us fix the spatial model that we had earlier and allow us to capture the information that this part of the image is more likely to be this, that part of the image is more likely to be that. They're usually constructed by intelligently averaging a bunch of training data. So we take a bunch of labeled images, average them together, align them together in some intelligent way, and that gives us an atlas. Now, one thing that you can also do is directly use the training images. And this is what people actually do in multi-atlas segmentation. So here, we register each training image. To our image. And then we're going to fuse their information using some kind of voting. Well, we'll say this image, once we registered it, said it was that. That image said it was CSF. This image said it was gray matter, and so on. And so if most of the images say gray matter, then it's probably gray matter. Notice that this is more expensive than single atlas segmentation. If our training set is large, we have to do a lot of registrations. On the other hand, this captures a lot more diversity within the images. Another way to do spatial modeling when dealing with a specific organ or structure is shape analysis. For example, here's a 3D model of a lung that's been segmented from a CT image like the one I showed you earlier. Shapes can be represented a few different ways. The simplest is probably with a set of landmarks. These are manually defined points that usually have some kind of anatomical significance. For example, on this lung here, the landmarks might be on the top of the lung, at this corner, maybe a few points along the bottom, and a few particularly relevant points along the center as well. Another way is through skeletal representations. For example, the medial axis transform represents a shape, either a 3D or a 2D shape, using a single line that goes through the center. A higher dimensional representation are S reps and M reps. These represent a shape using a plane that goes through the middle, as well as sort of what you need to go out from the plane to get to the surface. Another common representation is using level sets. So a level set is basically a function that's 0 on the boundary of the object, or the shape that's being represented, positive inside, and negative outside. And having this function is kind of nice because then you can combine different shapes simply by having a linear average of their functions. And working with the space of functions is usually quite nice. One commonly used level set representation is the signed distance transform. And this is really just the physical distance from the boundary, but positive inside and negative outside. So this location here, if this were, say, 3 millimeters, then the value at this voxel would be -3. Similarly for a voxel 3 millimeters inside from the boundary. So once the representation is chosen, the shapes can be modeled using something like PCA or any other way to represent the variability in the shapes. Sometimes the representations themselves are modeled explicitly, and other times they're used as a better spatial model for some kind of segmentation process like the one I described earlier. So this was a quick overview of some popular segmentation methods. It's impossible to cover everything in just 15 minutes, but hopefully now you have an understanding of at least a few popular methods. So some quick acknowledgements. The images and the data in this video came from these sources here: the MEC2014 Tumor Segmentation Challenge, the Buckner 40 Brain MRI dataset, and a Midas lung segments dataset. All the visualization was done using 3D Slicer, which is a freely available tool for visualizing medical images, as well as performing a lot of common medical image computing applications on them. For a list of review papers and references for the works described here, you can go to the URL here, and there are links to all the papers. Thanks for watching.